question I have that I'm pretty intrigued by after going through all of our data for the influenza and the Mycoplasma is what's happening at the Isle of Shoals. Now, the Isle of Shoals are a cluster of rocky outcroppings or rocky islands off of the coast of New Hampshire and southern Maine, where it's a mixing ground of gray seals and harbor seals of all age classes. And from working with some of our colleagues, the population there seems to be growing. So what is the effect of that growing population, the mixture of species, of age classes, that seems to be kind of stirring the pot of these infectious diseases?
I have been working with all of these people for a number of years now. Patty and I have been together since the early 2000s. Katie came a little after that, and the other Katie a little bit later. But one of the great things about working here is you get to meet and work with really smart, interesting, fun people. You become friends with all these great people, and you get to collaborate with them on great projects. So Patty's going to go first. Patty Schilling is a marine mammal supervisor here at the aquarium. She joined us in 2004, and she's been working with the northern fur seals, the species she's going to speak to you about, since they came here in around 2006. Patty received an internal grant here called the Cunningham Award in 2010 that allowed her to join a team from the National Oceanic and Atmospheric Administration doing fur seal surveys in Alaska, and she's gone back to do that annually since then.
Hi, everybody. So I thought I would start out just giving you a little bit of a background on who fur seals are, go into their current status, and then talk to you a little bit about the fur seals that we have here at New England Aquarium as well. So on the map, kind of shows you briefly where we are and where you would find fur seals out in Alaska. So I've had the pleasure, as Charlie mentioned, of working with northern fur seals here since 2006, and I've worked with a number of animals that have come through our doors and really fell in love with the species, who they are, and their unique personalities, and that really encouraged me to learn more about them. And so through that Cunningham grant that Charlie mentioned, I left Boston, Massachusetts, and traveled all the way to the West Coast. If you follow this map, it's a pretty long distance to find where northern fur seals would be located out in their native habitat. And so northern fur seals are found not only in Alaska, but actually 400 miles off the coast of Alaska on a set of islands called the Pribilof Islands. And so as you can see, they're pretty remote and fairly small. And the common question that we get here at the aquarium is, what is a northern fur seal? Which is a valid question, because unless you're out in Alaska on a cruise, you probably wouldn't see them unless you have the good fortune of being here at New England Aquarium. So northern fur seals are part of the Otariidae or Otariidae family, and that's defined by the fact that they are fin or feather-footed. They have the ability to walk on land. They can tuck their hind end right up underneath them and walk on all four flippers. They swim using those long front flippers at about 20 miles per hour is their max speed. And they also have pinnae, or ear flaps, on the side of their head, and this is handsome Cheetah showing off his ear flaps. But what sets them apart? Because those are all actually features that California sea lions share with them, and so oftentimes they're confused for a California sea lion. And the feature that really sets them apart is their fur. So this is a cross-section of northern fur seal fur. And what you can see on the top is guard hair, and underneath is underfur. If you make an OK sign with your hand, we actually do this in our training sessions at the New England Aquarium. They have about 300,000 hairs per square inch. If you look at the person sitting to the left of you and the person sitting to the right of you, and you put the hair on all three of your heads together, that comes to about 300,000 hairs in total. And they have that compressed into 1 square inch of space. So you can imagine it's pretty amazing, and that's how they're able to stay warm. So this is our northern fur seal Ursula in wet version. This is her as she starts to dry out a little bit, and this is what she looks like completely dry. And all through that process, she never has any water getting through to her skin, and so they don't have the need for blubber like a California sea lion or a lot of other marine mammal species, and that's really what makes them unique. It's also, unfortunately, what put them in jeopardy in terms of their population. So historically, the Pribilofs were discovered, or St. Paul and St. George, where they were found, were discovered in 1786 by Gerasim Pribilof. They were found pelagically prior to that, but their prime breeding areas, the area where they spend their summer months, is St. Paul and St. George. The native Aleutian population was sent there by the Russians somewhat forcibly to settle there and start the fur seal industry, the fur seal pelt process as a commercial Russian process. And so it was very unregulated initially. They were able to do it pelagically, which means they went out in the open ocean and they captured fur seals, oftentimes females, which left their pups on land, unfortunately, which did not survive. And then they also harvested animals on the islands as well. So in 1911, they passed the International North Pacific Fur Seal Treaty, and what that meant is that countries who were just helping themselves to fur seals for their pelts out in the ocean were going to stop that process, and the exchange was that the Russians who owned the islands at that time would give some of the profits to those countries in exchange. And so even at that time, people realized that these populations, they were not going to continue to reproduce without some kind of conservation measures. And that was the initial process of working towards conserving this species. So in the middle picture, you'll see that that's actually a fur seal dress that was created. It's hundreds of years old, and you actually can find it in the St. Paul Museum. And it's beautiful when you see it in person, and it's incredibly warm, I'm sure. And it was a great source of commercial income. The bottom picture is showing you some fur seal pelts. They were salted as a way to conserve the pelts. But what this did to the population was pretty scarring. And so you can see in the top graph here that this is pup production. And so in the 1940s, there was as many as 500,000 pups being born. And by 2015, there was less than 100,000. And so it made a really extreme impact what happened back then and to this present day. There was believed to be as many as 5 million northern fur seals back in the 1700s. There are currently about 600,000, so approximately 10% roughly from the time when fur seals first were hunted. Now, fortunately, fur seals are part of the Marine Mammal Protection Act, and they're no longer being hunted. The commercial fur seal industry ended in 1984. But to give you kind of a reference of where animals were, if you look at the bottom left graph, or left picture rather, that's an area called Paulovina Cliffs, which is a rookery on St. Paul Island. And at one point, it was teeming with fur seals. These animals were so prevalent that they were up over the top of these cliffs, which are incredibly steep, so they had to climb pretty high in order to find a space where they could find their own little nook. And the picture to the right is 2005, and you can see it's pretty barren. There's very few animals, and it's pretty unfortunate to see that. So I actually had an opportunity to go and work out there, and they put you in this really fancy attire. So it can rain there, and 15 minutes later it can be sunny, so you wear rain gear. You also have really long gauntlets, they're called, because you work with fur seal pups, and I can tell you in person they have incredibly sharp teeth. And so when you're working with them, you actually give them what I call pup haircuts. And so we mark a section of fur that I'll go into in a little bit, and so you wear those gauntlets just to protect your arms. So to do this research, you go out on the rookeries, and there are a number of them on both St. Paul and St. George, and we mark a certain number of these pups. And I struggle with my dad. Usually about every 6 months we go over how this process works, and I've yet to really communicate it with him. So I'm hoping tonight I do a better job with you guys. So the process is called mark and recapture, and what that means is we go out and mark a certain number of animals, and the scientists go out and recapture the data. So in order to recapture the data, scientists go out and stand up on these tall tripods, as you can see in the top left picture, look down below them at all of these animals, and they try and count the number of pups that have been marked by having that section of fur removed from the top of their head, as you can see in the right-hand picture, as opposed to the number of animals that have not had fur seal or had fur taken off. Now, if you look on the bottom two pictures, it's impossible to count how many little animals are all in there, and those animals are in motion. So that happened to be a still photo, but imagine that those animals are constantly moving around, shifting, going in the water, coming back out, and that's what those scientists have to deal with. So to give you an idea of how this process works, for example, if 100,000 pups were supposed to be born 2 years prior, we take that number, we hope that that same number of animals were born this year, and I actually am going to have the opportunity to go this year and work out there in August. So the scientists will tell us, okay, if there was 100,000 born last year, you're going to mark 10% of that, so 10,000 pups this year. So with that thought in mind, will go out and do that process. So this is a group of 20 fur seals. When the researchers go back out, they happen to see in this group there's 2 animals who are marked, those blue fur seals, as opposed to the 18 animals that are unmarked. That's a ratio of 1 to 9. And that's exactly the ratio you would want if you saw a population that was continuous, that was the same as 2 years prior, which would be great for the northern fur seal population. We would be thrilled if we saw that. If this is the ratio you saw, if there was only 1 animal marked to 19 animals unmarked, that meant that the population was increasing. And that would be even better news for us. That meant that we marked less than 10% of the population was out there. We actually only marked 5% of the population, and so there was a population boon. Unfortunately, what we tend to be seeing is this, that there are far more animals marked than the ratio that we are expecting. Where we are hoping to see 2 out of 10, we're seeing 4 out of 10, or in this case, there's 6 out of 20, or 3 out of 10 animals that are marked, and that means that the population is continuing on a decline. And that's the unfortunate current state of the northern fur seal. And this chart that Katie actually put together, she was kind enough to let me use, and this shows you where the fur seal pup production numbers are for St. Paul Island. So where they were once incredibly high and heading upwards in the 1970s, this you can see where they currently stand around the year 2015, and they're heading towards around 50,000 a year. But we don't want to end it on a gloom and doom kind of note. There are things that we can do that are positive for the northern fur seal population. They may not be direct, and that's a question that we often get is, well, what can I do? So the first part of the questions we get after our training sessions is, who are they? And the question we get towards the end of a conversation is, what can I do? And there are a lot of things that you can do that indirectly will affect the northern fur seal. So we're going to bring it back to Boston, and I'm going to share a little bit about the animals that we have here so you can understand why we have northern fur seals in Boston, thousands of miles away from Alaska, and how these animals really have an impact that far exceeds anything that we would expect for these animals. So we have Ursula, Cheetah, Luna, and Kentovi in our current fur seal collection. We have 1 male and 3 females, and we hope that this actually will become a breeding group in the foreseeable future. And we have a program starting up this year, Life According to Fur Seals, and we hope that you come and enjoy our presentations and learn more about our animals as well. And really this gives us an opportunity to meet with people, speak about our animals, and share their information. And more than that, it enables us to empower people about what you can do in your daily lives to change our behaviors that might impact fur seals, not directly, but indirectly. And it might be something small, say that you bring reusable bags to the grocery store that day. Say you take a walk rather than driving your car somewhere that you need to go for that afternoon. Those things may not directly impact a fur seal out in the wild, but they will have a profound impact eventually. The things that fur seals are dealing with out in the wild are beyond our scope that we can handle right now. They are things like overfishing that happened years past, so that now fur seals are dealing with issues where pollock, which is the main fish that they eat, is pretty low in calories, and that's because of overfishing generations behind us. We're wiser to that now. We know that we've made some mistakes, and so pollock is being sustainably fished, but it's still serving a detriment because that pollock is a kind of empty calorie fish for the fur seal. So we can change things now. We can eat sustainably in terms of the seafood industry. Climate change is a huge issue. Obviously, we've all heard it. There are things that we can change now that might have an impact on fur seals in years to come and with the generations to come. So what's happening to fur seals that's pretty unique is that they're stuck on 2 islands for their breeding season. There's nowhere else for them to go. And so when the weather warms up and the fish move away, they have to swim further to find food. They don't really have a choice. And so those females come back to the island, perhaps with a little bit less in their system to feed their pups, and that affects them. But I just wanted to show you kind of the impact that these animals have, our northern fur seals here at the aquarium have on people, and the impact that then we can have in sharing this message of it's not too late to make changes here in our neighborhood in small ways, in civic ways. You can get together with your neighbors and make changes in your neighborhood on things that are important to you, whether it's changing the energy usage you use in your community, something that's a larger project that you've had waiting in the wings in your community. These things can happen and they can make an impact. So just run through this really quickly so you can see these guys. my favorite picture. This is Luna, one of our youngest fur seals. And this is a group of students coming into the building. They haven't even made it into the building yet, and she's already captivated their attention. We're in there in the morning. It's probably a little bit before 9 a.m., and she's just checking them out, and they're checking her out. And she's already made that connection without even knowing what she's doing. And it's just pretty profound, the impact that she's had on these students before they've even joined the aquarium. So I just wanted to thank all of these people for supporting me, for being a part of the Fur Seal group, and just wanted to say thanks. And Ursa's going to blow you guys a great big kiss. Thank you, everyone.
Thanks. Okay, next is Katie Graham. Katie's an assistant scientist here in the Anderson Cabot Center for Ocean Life. She is an endocrinologist, so she studies hormones in a variety of species. She just sent us all a paper about her former work studying reproduction in frog species. But now here at the aquarium, she's mostly studying things like seals and sea turtles and whales. So Katie really brought in some tools here that have turned out to be very useful for studying hormones in wildlife. So she'll tell you about that.
Thanks, Charlie, for that introduction, and thanks, Patty, for really setting the stage and giving us kind of an overview about fur seals. It allows me to dive in and tell you a little bit about the science we're doing using feces to study our seal species here at the aquarium. I have some fun titles throughout the presentation, so keep an eye out for those. So this is me on my first day of kindergarten with my pigtails. I'm ready to take on the world. And like a lot of kids when I was little, I thought about what I wanted to be when I would grow up. I thought about being a teacher. I thought about being a doctor. But never in my wildest dreams did I imagine that I would grow up to study feces. But here I am. And all poop jokes aside, feces is actually a really important tool that we can use to learn a lot about wildlife and as well as ourselves. It's kind of a treasure trove of information. And for me, the compounds I'm most interested in studying in feces are hormones. So I'm going to tell you a little bit about our work with that today. And this is actually a tool I've been using for almost 10 years now. Starting out when I was in college, I actually worked at the Oregon Zoo, and I did a study looking at stress hormones in lions as they moved into a new enclosure. After I graduated from college, I worked for a few years at Disney's Animal Kingdom, where I had the opportunity to use feces to study reproduction in a variety of different terrestrial animals like giraffe and rhinos, even traveling all the way to Colombia in South America to study cotton-top tamarin monkeys in the forests. Like Charlie mentioned, in graduate school, I worked with critically endangered frogs at the Memphis Zoo. But I took a little break from feces for that work, but ended up finding myself using urine to study hormones in these animals. So I seem to not be able to get away from excrement. And finally, about 2.5 years, I started here at the Anderson Cabot Center for Ocean Life, where I have the opportunity to use feces to study reproduction and stress in a variety of different marine animals, including the northern fur seals here at the aquarium. And what you'll notice about all of these different positions that I've held is that I've been able to work either at or with a zoo or aquarium. And to me, this is something I'm really passionate about because I feel zoos and aquariums are incredibly important conservation research institutions. And I hope that's one of the takeaways that you'll get from our talks tonight here. So the program I work under is called our Marine Stress and Ocean Health Program. And this is housed under our Anderson Cabot Center priority area of marine animal and ecosystem health. And really the goal of our program is to identify and characterize different threats impacting marine animals and their habitats. And this program really got its start about 20 years ago now, when my supervisor, Dr. Roz Rowland, started using fecal samples to study the health and physiology of right whales. And this is a lot of incredibly important data that's been collected over the years. And so data like this, along with some from our other projects, have been used to do scientific publications and generated data that's been used to help with mitigation and other conservation measures. So we really have applied science here. But before I delve into the specifics of my project, I wanted to share just a little bit about what a hormone is, because we throw this word around in conversation. We joke about hormonal teenagers and stress hormones in terms of our jobs and whatnot. But to give you a definition of what a hormone is, you can think of them as little chemical messengers that circulate in our body that control lots of different biological processes. So they're produced by different glands and tissues in our body. They travel in the blood, and then they move to different areas of the body where they stimulate responses in those tissues. And while they have a variety of different functions, the hormones I'm most interested in are reproductive hormones. So hormones like progesterone, testosterone, and estrogen that you've probably heard about in relationship to your own body. And those are actually the same hormones that we're able to study in our northern fur seals here, as well as a variety of other animals that we're studying. And these reproductive hormones control processes like pregnancy, estrous cycles, and puberty. And while we could study these hormones using blood, just like your doctor probably does when you go get your annual physical, you may get your blood drawn and they may measure hormone levels, we actually find that feces is a really valuable tool for us because it can be collected non-invasively. So what happens is as those hormones are circulating in the blood, after they've relayed their message, the body then breaks them down in the liver and the gut, and they ultimately get excreted in the fecal sample. We can then collect that fecal sample and study all sorts of different aspects about the animal from it. And really the value with the fecal samples is that we can collect a lot of samples over time since it's not impacting the animal's welfare or behavior by collecting these samples. It's a non-invasive measure, which makes it really powerful in terms of improving animal welfare when we're trying to do these types of studies. And these fecal samples really represent sort of an accumulation or an average of the recent circulating hormone concentrations. So we get an idea about what the blood hormone levels were like recently with this. And while fecal hormone analysis has been used for a variety of different terrestrial species and more recently other marine mammals, it hasn't really been applied before to northern fur seals. So that was one of the reasons we wanted to take on this project. And we really, we have a lot of baseline data from this because other groups have studied blood hormones in northern fur seals, but we're hoping this new technique will allow us to do this non-invasive kind of study. Part of the reason we wanted to study reproduction in northern fur seals is because they have a very complicated reproductive strategy. To put it scientifically, they're weird. And I'll tell you a little bit about why that is. But also, as Patty highlighted, these animals, many of the populations are facing decline. So having more tools available to study their reproduction is a really important thing. So some of the quirks that fur seals have in terms of their reproduction is that they're highly seasonal reproducers. What I mean by this is each summer, females come to land, they give birth, and then within just a couple weeks, they've actually bred and gotten pregnant again. So they're really tied into the seasonal and environmental cues so that their pups are being born at the proper time each year. Females also exhibit something called pseudopregnancy, and this is when the hormone levels in a non-pregnant female look very similar to the hormone levels in a pregnant female. And this can make it very difficult to diagnose pregnancy in the species, and it's definitely a challenge we're interested in taking on. Females also show something called delayed implantation. So if a female is pregnant, shortly after fertilization, that little embryo starts to grow into a small ball of cells, but then it stops growing, and it just floats around the uterus for several months before it finally then implants and begins to grow again into a fetus and then a pup. And lastly, for males, the age of sexual maturity isn't really well understood. In the wild, only the biggest, toughest males are usually the ones that get to breed, so we're not quite sure when males are actually reaching sexual maturity because usually only the bigger, older ones are getting to do that breeding. So given all of these quirks and just some of the general questions and unknowns about fur seal reproduction, our marine mammal team here actually approached us about trying to do this study. And partially because they wanted to learn more about the animals, but they really wanted to learn more about their animals so that they could help take better care of them. The data we can provide through our fecal hormone analysis may allow them to know more about their animals, providing them the optimal care. And a lot of our study questions are actually based around these gaps in knowledge. Things like the age of sexual maturity in males. We have that young male here, Cheetah, who's kind of on the cusp of puberty. And so for us, it's really important to know if and when he's entering puberty, as that may affect how he interacts with other seals, as well as how he interacts with his trainers. As I mentioned, pregnancy is also very difficult to diagnose in this species, so we're hoping that we can use fecal hormone analysis as a way to get an early detection of pregnancy for the species, allowing us to get mom and pup the best care. And because this data is valuable not just to us here at the aquarium, but to any group that's trying to work with northern fur seals, we reached out to the two other aquariums in the US which house northern fur seals, Mystic Aquarium and Seattle Aquarium. And we invited them to participate in this study, so they're actually contributing and sending their samples here for analysis as well, which is really cool. It's a really collaborative effort. But because this is kind of a new study and hasn't been done before for fur seals, we had to start out with some really basic questions, like can we even collect a fecal sample? And whose poop is whose? So it can be, if you've been to our marine mammal area, you know it can be challenging potentially to find a fecal sample in there because we have not only multiple fur seals in there, but also the sea lions. So in order to figure out who was leaving samples behind, we had to come up with a creative solution. And that solution was actually fecal markers. So fecal markers are unique natural diet items that are added into the fish that are fed to the fur seals. Items like sesame seeds, lentils, barley, and rice. Just like there are some items in our diet that don't get fully digested, the same principle holds true here. And so these items get loaded into the fish that are fed to the fur seals. The fur seal eats the fish, leaves our sample behind, and then our very dedicated marine mammal trainers, thank you guys, collect our fecal samples, and they look for those fecal markers so that they can then match them up to the individual seal. It allows us then to label those samples and send them up to the lab so we can get individualized samples on those. Once in the lab, it actually takes a lot of work for us to get to the point where we can do a hormone measurement. We have to freeze dry our samples, it sucks all the water out of them. We then sift and weigh, and if you actually look closely in the middle picture, you can see those fecal markers. They get sifted out there. We then take that fecal powder and we extract it using alcohols to turn it into a liquid, which we can then measure using our tests. And you can think of these tests, they're called immunoassays. You can think of them like a really fancy at-home pregnancy test. So we load all of our chemicals and our samples into these little tiny wells on the plate, and we get a color change reaction depending on how much hormone is in the sample. Basically, if you have a really light-colored well, that means there's a lot of hormone in the sample, whereas if you have a very dark-colored well, it means there's not very much hormone in the sample. We can then quantify these patterns and start to analyze them to look for patterns. So thus far, we've been able to do a little bit of collect a little bit of preliminary data for this study. And what we found so far, for example, is that females tend to have much greater concentrations of progesterone compared to the males. We also found that adult males tended to have a greater concentration of testosterone compared to the juveniles. So this may be a good indicator for sexual maturity in the species for us. What we're hoping to do in the future is really provide sort of in-depth looks at each individual animal within the aquarium population. We want to track these hormone patterns over time throughout the year, allow us to really understand what's happening with these animals, and also maybe start to identify some of these markers that may be a good indicator of these different reproductive events. So I hope what you'll take away from this talk today is that, first of all, aquariums are very important conservation research institutions, and they have a great opportunity to study the animals under their care. I hope you've also learned that feces can be a very valuable, non-invasive tool, and that the data from these studies can benefit management, conservation, breeding programs, and just the overall care that an animal receives. For northern fur seals, we have some early evidence that fecal hormone analysis may be a valuable technique for us to learn more about reproductive patterns in the species, and we could use similar type studies on other seal species in the future if we wanted to do that. But ultimately what we'd hope to do is take this type of study and apply it to, or take this technique and apply it to study northern fur seals in the wild. As these populations are declining, it's important for us to have tools to study them, and we could potentially add in other hormone biomarkers like stress hormones or metabolic hormones so that we could assess not only reproduction in these species, but also address some of the human impacts that may be threatening these groups. So with that, I'd like to say thanks to the many people that contribute to this study. This is really a collaborative effort and would not be possible without the input of many people. So I'd like to thank my marine stress and ocean health team, particularly Roz Rowland and Liz Burgess, Kathy Streeter and our marine mammal team for being tireless advocates for this project and enthusiastic sample collectors. Thank you guys. Charlie Innes and our animal health department, our collaborators at Mystic Aquarium and Seattle Aquarium, Tracy Romano and Sean Larson, and lastly, the Epley Foundation for Research, who provided some of the funding for this initial study.
Thanks, Katie. Next is Katie Poliaris-Bonner. Katie's been with the Greater Atlantic Marine Animals Stranding Network for the past 12 years, and she's been working with us here at the aquarium for almost 8 years now. She has conducted necropsies, which is an animal autopsy on over 500 marine mammals, including large whales, dolphins, porpoises, pinnipeds, and also sea turtles and penguins and other marine birds. Katie, in addition to doing a lot of the necropsy work for us, is a critical member of our sea turtle rescue rehab team down in Quincy. So seasonally, we sort of put the hold on doing a lot of marine mammal necropsy work when we transition over to all of the sea turtle rehab work that we do. Katie was really key here. She was the person here in 2011 that noticed something was a little bit amiss with some seal mortalities that we were seeing along the coast here. And through her work a brand new form of influenza virus was discovered that had migrated from bird species into seals. So that was an impressive piece of scientific work. Required a lot of collaborators, and it was all from Katie's keen insight and thoughtfulness that really led to all of that. All right, ready? Thanks.
Well, thank you, Charlie. You just gave my talk. Well, good evening, everybody. Thank you for giving me the opportunity to share with you the efforts of the rescue department here at the New England Aquarium, in particular to share with you what we do with the data collected through our necropsy program. So many of you folks may know about the Rescue Rehabilitation Program, mostly due to our efforts with working with sick and injured sea turtles, particularly the Kemp's Ridley sea turtle. Every year, every November and December, cold sun sea turtles strand along the shores of Cape Cod Bay in the hundreds. And our partners down at Wellfleet Bay Audubon Society walk the beaches at every tide to find these debilitated sea turtles. They bring them up to our facility in Quincy, to our hospital facility, where we provide clinical care for them, we rehabilitate them, and then we release them back out to the wild. But I'm not here to talk about turtles. Marine mammals are finally going to get the limelight today. The marine mammal rescue team here at the New England Aquarium has actually been around for one of the longest stranding networks in the country. We have records of stranded animals dating back all the way to the early 1970s. Currently, we have 5 full-time staff, 1 part-time staff, and an awesome crew of dedicated volunteers. And we respond to all reports of live, entangled, wayward, injured, sick, harassed seals from Salem, Massachusetts, south through Plymouth. We also respond to porpoise and dolphins that are caught on land with outgoing tide. And then mostly, though, we respond to dead animals, even large whales, as you can see here. To be more specific, we respond mostly to dead pinnipeds. We have the gray seals and the harbor seals are residents here year-round. We also have seasonal visitors such as the harp seals and the hooded seals. From the dead animal data we collect, it's really important information to help us understand the populations that we're working with and to understand the distribution and the species within our response territory. And I know it doesn't sound as glorious and rewarding as saving the life of a baby seal. However, the information that we do collect does eventually save the baby seal, which I'll show you a little bit later. So what's the big deal with information from dead animals? Well, most importantly, it's required by law. We are authorized through NOAA fisheries to respond to marine mammals. All marine mammal species are protected species, and we have this authorization from NOAA to respond to animals live or dead. And within that agreement, we share with them the data that we collect, such as the species, the location, the gender, and the age class. Also, that information helps us get a better understanding of baseline information on the population and the distribution of species within this territory and within the greater Atlantic region. When we get to that animal on the beach, and if the carcass is fresh enough, meaning that it's 48 hours to 72 hours post-mortem, we'll bring that carcass back to our facility in Quincy, and we'll do a complete, comprehensive necropsy examination on that animal. Or what Charlie was explaining, a necropsy is an animal autopsy. Through the necropsy examinations, we learn about the biology, the anatomy, the physiology of the species. Some information that we know about marine mammal species come only from stranded animals. So you can see how the information we collect from the dead animals is very important. We collect samples from the tissues to help us identify the presence of disease and to determine the cause of death, which then can be used to inform and improve our clinical care of the live animals we respond to. So see, this information does eventually help that baby seal on the beach. When the data are consistently collected for several years or more, we now build a robust data set of pathologies of these animals, which then can contribute to any investigations in population health and ecosystem health. Monitoring the dead animals as well helps us keep an eye out for any emerging, resurging, novel diseases and mortality outbreaks. We also investigate every carcass for the evidence of human-caused injury or mortality. So all the data that we collect from the animals that we necropsy are similar to solving a puzzle. And I am a big fan of puzzles. So if you think about it as the individual animal, you have all these pieces. You have the behavior of the animal if you see it on the beach. You have any clinical information, blood work. You have what you get from the gross observation. So gross is what you see with the naked eye. You have your information on your pathology reports and your ancillary diagnostics. all those little pieces help solve the puzzle as to what caused that animal to die. And then when you look at a greater picture, each individual that we get to look at that detailed are tiny pieces of the larger population, a larger ecosystem puzzle. So as Charlie just explained, I will briefly, I guess, go over this, or maybe in a little bit more detail. Our first puzzle to solve happened in 2011 when about 170 or more harbor seals showed up dead along the shores of southern Maine and northern Massachusetts within a relatively short period of time. So I'm going to tell you the story of the seal flu of 2011. It all started on a warm, rainy day in September when we received a report of 4 fresh carcasses in one day on the tiny coast of New Hampshire. Seemed a little odd, but really nothing too alarming. However, the very next day, we received reports of 8 additional carcasses, 6 of which one of our biologists here, Carrie McNally, called from the beach and said, there's not just one seal here, there's 6 seals here on Jennings Beach in Rye. That was a light switch for us, for sure. Usually, stranded animals will come on shore, or the dead animals will wash up because they have a chronic condition happening. Whatever their disease may be may cause them to decrease their feeding efforts, and then a lot of the times they're emaciated as well. What we're seeing here is not only the spike in the number of animals in a small geographical area, but also how these animals presented. They were all harbor seal weanlings, and they were all relatively fresh, meaning that their time of death was recent, and they were all in good body condition. So usually when we see a dead animal that is in good body condition, but is not alive and has no external reason for death, then we start to think of something suspicious, infectious disease or others. So we started to work with, so just to give you an idea of the difference in the numbers and why that would seem so significant for us. This figure here, the bar graph, shows our 5-year average of what the number of animals that we responded to on a monthly basis from 2006 to 2011. So this is a 5-year average by month. This graph is the same bar graph, so the bar graph is 2006 to 2010. We then put on the data from 2011 on top of that, which is the orange line. And you can see in some of the months we had 3 to 4 times the number of dead seals stranding within our region. And during the peak of the stranding period, we collected samples from about a dozen fresh carcasses throughout our response area. And the common denominators that we found included that they were all weanling harbor seals, all in good nutritional condition or good body weight. They all presented with oral ulcerations as well as dermal ulcerations, and they all showed evidence of pneumonia, whether it be emphysema, edema, or necrosis. We shared these gross findings with our veterinarians and with our pathologists, and from there we started to go through a process of elimination of what could possibly be the cause of this very fast, rapid death of multiple animals. And we based this process of elimination on our prior knowledge of the species, of our ecosystem, and other epizootics that had happened locally and globally. So some of the things on the top of our list were, of course, the effects of a harmful algal bloom in the area. It's not foreign to the New England area. So we started to work with our collaborators down at Woods Hole. We worked with the state departments. They did plankton toes, they surveyed the shellfish. We submitted samples, tissue samples from our seals, and everything came back negative or below reporting levels. So we can knock off the harmful algal bloom off our list of the etiology. In parallel with that, we also submitted samples for Morbillivirus. Morbillivirus is a common virus, respiratory virus that circulates through populations and can cause an epizootic, such as over in Europe, it killed tens of thousands of seals. So we sent in those tissues, and that came back negative, so it wasn't Morbillivirus. Also on our list was EEE, or West Nile virus. So these are some histology slides. These are respiratory tissue, but you diagnose EEE and West Nile virus by looking at brain tissue. And so there was an absence of non-suppurative encephalitis, so we crossed those off our list. But here on these slides, you'll see this is respiratory tissue. What we did find was diffuse pneumonia with a 95% loss of air exchange space within the tissues. This area is flooded with inflammatory cells associated with a bacterial infection. And the mucosal lining is lined with small bacteria, what at that time the pathologist suspected was Mycoplasma. But most importantly, there was evidence of hemorrhagic alveolitis, which often is associated with a viral infection. So from here, we started to do some big-time virus hunting. We worked with a collaborator down at Columbia University. We sent a suite of samples from our individuals, and we looked for a whole bunch of viruses. And what came out as the heavy hitter was the influenza. And it was an influenza A and a novel strain, H3N8, that's never been documented in seals before. And this we found in all of our seals that we sampled. And something to keep in mind also is that Mycoplasma. So Mycoplasma phocidae is a small bacteria that lacks a cell wall, and it's a pretty aggressive bacteria because of that reason. So we did find Mycoplasma as well in all the seals that we sampled, that we submitted. So to tell you a little bit of information about what we learned about this novel strain, the H3N8, we did full genome sequencing, which demonstrated that the closest relationship that this particular virus had was to North American waterfowl with no obvious reassortment, suggesting that there was a direct transmission from wild birds to the seals with some evidence or some level of seal-to-seal transmission. In a laboratory setting, this particular virus did not produce clinical signs in the birds or cause death. However, clinical signs were obviously evident in our seals, as well as certain mutations of the virus showed that there was increased pathogenicity. Staining of the lung and the tissue on your far left shows that there's a host-mediated response to the infection in the presence of viral antigens. And then also the darker pictures here show that there's confirmed presence of the influenza virus actually within the tissue, and they clustered around the trachea and the bronchioles within the lung. So there is active virus within the lung tissue. Now, this outbreak is particularly significant because the virus has naturally acquired mutations known to increase transmissibility and virulence in mammals. Monitoring the spillover and the adaptation of avian viruses in general into mammal species is really important if we're to understand the factors that enable the emergence of new pathogenic and epizootic and zoonotic influenza viruses in general, especially for implications for human health. So who would have thought that one day, with Carrie McNally walking the beach, finding 6 dead seals within a quarter of a mile would turn into something that informs the greater public health as well. So, oh, maybe I didn't show you guys that. I meant to do that. I'm sorry. So these are some of the publications that have stemmed from the virus that we found. You can see a lot of people are interested in influenza because of the way that it mutates so rapidly and how it can transfer from animals to people, which is called zoonotic. And this is some of the literature that's been published from our strain of virus that we found. So on to our next puzzle, and this one stemmed directly from the influenza investigation I just shared with you. But remember how I mentioned that little bacteria, the Mycoplasma? This is where that comes into play. I didn't show you there how often that little bug was found in our seals. I said it was found in a lot of them, but what came from the influenza study really triggered our next phase of this investigation. So this data comes directly from that influenza study, and you can see here the map where we have the Mycoplasma influenza co-occurrence rate we found in 6 animals within that study. Interestingly enough, we also found Mycoplasma alone in an additional 4. So if you count up the total number in this data set right here, there's 13 animals. We found 10 of the 13 positive for Mycoplasma within the respiratory tissue, but only 7 of the 13 had flu. So what is the significance of this Mycoplasma? I was really intrigued by it because although the cause of death and the cause of this unusual mortality event was determined to be this novel influenza, no one ever really fully dove into the effects of the Mycoplasma here. So we wanted to really investigate, who, what, when, where, why, and how of this Mycoplasma bacteria, and get an idea. Our goal of this project was to get baseline information on the presence and the presentation of Mycoplasma in respiratory tissue and disease of New England seals. And thanks to the Van Sloan Foundation, who provided some funding to embark on this very small project, but it was a very productive project as well. So just give you a small background of our regional history of Mycoplasma in Feces, especially of New England. What we found in our 2011 UME was not unique at all. So back in 1979, 1980, a similar occurrence happened here where it was an epizootic, a large number of seals died. It was the same geographical region, happened to juvenile harbor seals. It was caused by viral pneumonia by a novel influenza virus. Interestingly enough, in the literature that has been published, the authors did say they suspect that there was a concurrent respiratory infection with Mycoplasma, but that's where they left it. They didn't investigate it anymore. Another thing that they mentioned was that likely contributed to also the perpetuation of the virus was the population density of harbor seals at the time, as well as the unseasonably warm temperatures, which is something that we saw as well. So we started to look into our data set. We had a 10-year data set, no, it was a 10-year, 8-year data set from 2011 through 2017. We submitted samples from 90 animals. We had representation from Maine, New Hampshire, and northern Massachusetts. We were able to submit samples from harbor seals, gray seals, and harp seals. All age classes were represented aside from dependent animals, although our sample set was largely dominated by weanlings. We were able to submit samples from all months of the year to try to figure out if there's any seasonality, although the majority of our samples were collected from August to October, which is opportunistic from when the animals do die during that season. So some of our results shows that we did find Mycoplasma within the respiratory tissue of 39% of the animals that we sampled. We found it in all years of our study except 2013, and I haven't dove into that yet as to what that might mean. Seasonality, there definitely seemed to be a little bit of a seasonality here, with 56% of the animals sampled in the summer, 43% sampled in the fall were positive for Mycoplasma within the respiratory tissue, although Mycoplasma was found in all seasons. Considering species and age class, it seems to be that Mycoplasma was present in mostly harbor seals and harbor seal weanlings, although gray seals of subadults and adults did have Mycoplasma as well. Interestingly enough, though, the harp seals were negative all around. When you look at the geographical location, again, another similar pattern as we saw in influenza. We did find Mycoplasma throughout our whole sampling area from the southern Maine area down through northern Massachusetts. However, if you zoom in there, this is on your right-hand side, that's the zoom-in focused area of the tiny little coast of New Hampshire. It's not many miles compared to Maine and Massachusetts, but we did find a large cluster of Mycoplasma-positive animals in that one little area. And I wanted to point out the Isle of Shoals on that map and how close it is to the New Hampshire coastline. Another great finding, not only of who it was in, such as the juvenile harbor seals in the New Hampshire area during the late summer and early fall, but is the variety of Mycoplasma we found. Now, this was an extremely unexpected find. Prior to our research, there were only 3 different types of Mycoplasma identified and published in Feces globally. Those are the ones highlighted in yellow. We found more than 17 phylotypes in our seals in New England, which is a really interesting finding. The other fun thing that we found was the number 1 listed here, which is the human Arctic Fascinoma, and you can see that we found it in 16 of our animals. It's the most frequently cultured species of Mycoplasma. This specific species of Mycoplasma was identified first in 2015. An Alaskan subsistence hunter was harvesting a ring seal and cut himself while harvesting the seal. That quickly turned into an infected wound with a disseminated septic arthritis caused by Mycoplasma. He did survive, but it took them a while to figure out that it was this Mycoplasma, which is an aggressive bacteria. There was only 1 case of this one type of bacteria, and we found it in 16 different animals on the completely other side of the continent in a completely different ocean. So just to show the variety of bugs that we have in our animals that are also zoonotic. So what is the effect of the Mycoplasma or relationship with respiratory disease in our seals? We found out who it was in, we found the different types of Mycoplasma, but what is its relationship with respiratory disease? So of the 90 seals that we looked at, the primary and secondary cause of death was attributed to bacterial respiratory disease in just 21 of the 90. Of the 21 animals that died from respiratory distress, only 8 of them were positive for Mycoplasma within the respiratory tissue. And these are images of what some of those animals looked like. The gross presentation was intralobular emphysema, mild tissue necrosis, interstitial edema, as well as congestion and hemorrhage. The remaining 27 positive Mycoplasma animals, so remember we had 35 individuals that were positive or cultured positive for Mycoplasma of the respiratory tissue. Their cause of death was sepsis from a traumatic wound, septicemia, encephalitis, parasitic pneumonia. So what this tells us is, what we already knew before is that Mycoplasma exists in seal populations. It's something that they carry in their mouth, but they don't show clinical signs of disease. They just carry it in their system. But possibly there's something that happens to them, something else that comes through their bodies, causes them to be a little bit more compromised, that allows the Mycoplasma to set into the respiratory tissue, which then causes this respiratory disease. Because although Mycoplasma was found in the tissues, or cultured from the tissues, the lung tissues, not all of them died from a respiratory disease. So what are our next steps? Well, we want to fully characterize that new Arctic Fascinoma, but we want to show the disease that's being caused within the lung tissue of those animals that had the bacterial respiratory disease, similar to what we did with influenza, where we showed the virus within the tissue. The other question I have that I'm pretty intrigued by after going through all of our data for the influenza and the Mycoplasma is what's happening at the Isle of Shoals. Now, the Isle of Shoals are a cluster of rocky outcroppings or rocky islands off of the coast of New Hampshire and southern Maine, where it's a mixing ground of gray seals and harbor seals of all age classes. And from working with some of our colleagues, the population there seems to be growing. So what is the effect of that growing population, the mixture of species, of age classes, that seems to be kind of stirring the pot of these infectious diseases? All right, so last but not least, the other cool thing that we've done with our necropsy program is not necessarily with dead animals. We're shifting from dead animals to mostly live, and we're shifting from harbor seals to mostly gray seals. And this is to look at the prevalence of alopecia in gray seals. Now, this study came about by just talking with our staff and getting reports of gray seals on our beaches and noticing that some of them are losing their hair. So we thought that maybe there has been an increase, but we didn't have any statistical numbers to support that thought. So we wanted to strategically go through all of our data over a decade to see if this is something that's actually increasing in prevalence. So the goal of this study is not only to assess the prevalence of alopecia in gray seals, but also to standardize the description of the clinical presentation of gray seals. Just to give you a brief overview of what alopecia is, it's the partial or complete hair loss at the follicle or hair shaft for many mammal species. It's found in domestic and wild animals, bears, bats, deer, dogs, monkeys, all kinds. Anything with hair can get alopecia pretty much. The important thing about alopecia that we should know is that oftentimes it's an indicator of an underlying disease process, or the presence of alopecia can then increase susceptibility to infectious disease. So some of the common causes, and there's a long list, but some of the common causes in the variety of species can be infectious agents, such as a bacterial disease, a fungal disease, or viruses. Alopecia can be associated with the virus. Ectoparasites often cause alopecia, mostly in deer and moose, and also known in some pinnipeds. Allergens are found to be the cause in domestic animals. Nutrition, such as deficiencies and toxicities. It can be related to hormonal imbalances and stress. We were able to go through all of our photos. We had many, many photos of seals to come up with a standardized way to describe or classify the different degrees of alopecia. So we went from mild, which means that if an animal has a mild case of alopecia, there is loss of hair of less than 10% of their body, and usually there isn't any associated lesions or irritation. And then you go to the other side, the other extreme, which is severe, where there is loss of hair over greater than 50% of the animal's body, and oftentimes there's lesions or irritation associated. And then moderate is in between. So looking at the prevalence, we looked at stranding data from 2004 through 2013, so a decade long of data, not only from our animals within our stranding response area during that time, as you can see highlighted in blue, but also our collaboration with the International Fund for Animal Welfare, which responds to seals over Cape Cod in southern Massachusetts. We also collaborated with the Center for Coastal Studies in Provincetown. They do annual surveys throughout the season of the haul-out sites, the major haul-out sites for gray seals on Cape Cod. And so we looked at their photographic data to determine the presence of alopecia in those animals. So putting all the information together, we were able to accurately and confidently assess the presence or absence of alopecia in 374 stranded animals and 1,768 animals at the haul-out sites over time. So looking at the data demographics, like who our data represent, you can see here that our stranded data are most largely represented by the weanlings, so the gray seal weanlings, which is in the light blue, whereas our haul-out site data is most largely represented by the adults. Now, when you put this information together, all the stranded animal data and the haul-out site data, you can see that it's about 75% adults, and then the remaining quarter is divided between the weanlings and then the other immature animals, which is a common representation of normal populations. So what did we find in our data set is that 7.2% of our almost 2,000 animals had some evidence of alopecia. However, the degrees of severity varied, and it was pretty much equally distributed. We found that the stranded animals tended to be affected by the alopecia more frequently than the animals at the haul-out sites. When you look at it over time, that was our first question, is this happening more frequently? You can see that the occurrence of strandings has increased, as well as the prevalence of alopecia within that population, or within the stranded animal data. This is just stranded animals. And the way to read this graph is, for example, in 2013, we had 99 reports of stranded gray seals. And of that 99 number reported, almost 45% of them had hair loss of some degree. So you can see the number of strandings is increasing, and also the percentage of alopecia of those animals is also increasing over time. When you look at the seasonality, it seems to be a peak during the late spring and early summer. And when you look at by gender, there doesn't seem to be any difference. Males and females have alopecia at the same rate. When you look at it by age class, you can see that although we had a large number of mature animals within our data set, only 13 had the evidence of alopecia. Whereas our weanlings, almost 1/3, or a little bit more than 1/3 of the weanlings were affected by alopecia. So in summary, one thing I didn't mention as well is the alopecia we found mostly in living seals. So it was very rare that we had an animal that died that we could actually sample. So it seems to be associated not with high mortality rates, but maybe high morbidity rates. And so we also found that it affected 7.2% of our data set, almost 2,000 animals were in our data set. The occurrence of it happened to be centered around Nantucket and Cape Cod, which is expected because that's where the majority of the population are. It was mostly found in the late spring and summer months, and it seemed to affect young animals more frequently than adults. So we were able to assess the prevalence of this condition, and our next question is, and I'm sure you're wondering, but why? What's causing this? And as I showed you at the very beginning, there are a lot of different possibilities. So that would be the next phase of our investigation, is trying to uncover or cross off our list systematically what could be the etiology. So the first questions you wonder are, could it be infectious? And if it is infectious, could the high population density of gray seals be increasing the transmission rate? Or are weanlings more susceptible to any infectious disease that's causing the alopecia because of their developing immune system? And as I said before, weanlings seem to be more, the conditions seem to be more prevalent in weanlings. You can also consider the effects of their diet, and are there any diet deficiencies or toxicities leading to the alopecia? Is there a difference in age class? Are the adults outcompeting the weanlings for the proper nutrition? Are the weanlings physiologically limited to where they can forage for food, which then is causing them to be deficient in some nutrients that are important for proper and healthy pelage? So as you can see here, this puzzle is not yet complete. We got a couple different pieces to understand who it's affecting and when, and those little gaps there are going to be hopefully the etiologies as to why it's happening in our gray seal population. So our take-home message today is what I think is that the dead say more than the living sometimes, but also that the rescue rehabilitation program and NEAQ as a whole are more than just sea turtles and tanks to come in view on the weekends or during summer vacations. The research that we do here make us better biologists, make us more informed veterinarians, and our work, as you can see through Patty and Katie's work as well, and what I presented to you guys is far-reaching, far beyond Central Wharf. So I'm very thankful that you guys came here to learn a little bit more about the research that the New England Aquarium is doing, and I hope you learned a little bit more today prior to what you came in knowing. And thank you very much. Thank you.
Okay, we can take questions now, if anybody has. I'll try to repeat so everyone can hear. Yes? How did the 4 fur seals get here in the first place?
They all have different stories. Sorry, it might be cutting out from you. Ursula was born at the New York Aquarium. Luna was stranded as a pup off the coast of Laguna Beach in California. Sorry, guys, out a little bit. Cheetah was stranded as a pup. He was actually left on Alaska Fish and Wildlife doorstep at 7 days old. And Kitovi was born right here at the aquarium. She's Ursula's daughter.
Everybody hear that?
The question was, how do you collect whale poop? I guess I'll take this one. So really, that's my boss's specialty, but I do get to help out with that a little bit. Some of our fecal samples come from our right whale survey team, which go out every year in the Bay of Fundy off the coast of Maine, and they actually can opportunistically collect fecal samples. They'll just be out on the boat, they're doing surveys looking for animals to photograph them and identify them, and so they'll opportunistically come across fecal samples. For a few years, a while back, we actually had a dog who was trained to sniff out fecal samples, much like a drug detecting dog. But a lot of our samples now are just opportunistic. And because we've become so well known in this field, a lot of groups will actually now send us fecal samples. We'll get emails, somebody is excited, we find a fecal sample, can we send it to you? So they'll send it here for analysis.
Let me repeat. Go ahead.
Sorry.
Go ahead.
The question was about the influenza virus and how do we think it got from the birds to the seals. So one of the cool things about viruses and what they're meant to do is to find their next best host. And so viruses are often known to mutate pretty rapidly. And so that's what we think happened. In our lab studies, we were able to show, it wasn't us particularly, but our colleagues at Columbia University were able to show in their, like their chicken egg testings that they did, that the virus actually did mutate to be more adaptable to the seals. Oh, likely influenza is usually respiratory. I don't know, Charlie, if you have a better explanation for
No,
that.
the theory was that some of these wild birds that may have been carrying it were in geographic proximity to these seals at some point, some of these migratory waterfowl. And it's usually transmitted through aerosol infection by coughing or sneezing. The middle up top.
There's two big reasons we think. Can I borrow yours? There's two things we think. One is climate change. So as the oceans are warming, the fish that they're looking for are moving further from the islands. And so the mothers are having to swim further. They're coming back with less energy to feed their pups. And so those pups are less inclined to thrive. So that's one leading theory. And then the other theory is commercial fishing is taking the same fish that they're looking for. So both those factors combined are what they're struggling against.
I have a question about alopecia. I know that it's been observed in pinnipeds and terrestrial mammals, but I was wondering if there have been any findings on it affecting Vibrissae in seals, but also maybe even in cetaceans, or if there's just maybe a bit of a deficit there.
All right, the question was alopecia that we know that we find it in multiple mammals and pinnipeds, but has it affect the Vibrissae, which are the whiskers on pinnipeds, or any cetaceans? Because cetaceans are mammals, and one of their things that makes them a mammal is that they have hair at one part of their life stage, and they are born with little Vibrissae, but they fall out shortly after. What we've seen in the gray seals and in other species is that the pattern, oftentimes when it is severe, is focused on the back of the head and the neck and goes over the shoulders, and oftentimes leaves the animal with a ring of fur on the face and on the flippers sometimes. And so I haven't personally seen it affecting the Vibrissae. But also, too, what the Vibrissae and the fur are made out of are very different, because the Vibrissae are more tactile, sensory organs, pretty much, whereas the fur is more for insulation and streamlining and such.
Everyone's getting excited. Yes.
All right, that's a good question. How long did it take? So once we saw that, that first animal that showed up that looked in good condition. I can remember the dates. I don't know why I'm good at remembering dates, but it was September 28th. It was a Wednesday. And we had this one seal that showed up that was robust, and it wasn't alive. So we decided to pick that up. And that's where Carrie was going, to pick that animal up the next day. And then we found the 8 others. So right then and there, that week, we tried to process as many as we could. We started talking to our pathologists. We submitted the samples for the biotoxin analysis in Morbillivirus probably that week. Those answers of the negative came back within a week. And that's when we say, okay, well, what's the next step? And we started to figure out, is it the histopathology takes a little bit longer, because the process of getting the tissues onto the slide, the process of it is longer. So we didn't get the pathology results or in the cause of death and diagnosis of disease for about 2 weeks after the initial. And then that's when we saw that there was possible viral inclusions. It wasn't Morbillivirus, so then we submitted samples to Columbia, and then it was in November, so probably about a month. It was in November where we found out that it was an influenza A. And then from there, it takes time to determine, actually sequence it out to be the H3N8. So it does take some time. So then by the time we found out that it was influenza, things started to slow down. But within that peak, we were sampling and archiving, collecting, because we knew we had to find the answer, but it might not be immediate.
The question was, when we mark the fur seal pups by taking fur off of their head, does that affect their thermoregulation? It doesn't, because those pups don't swim until they're past the point where they get a new coat of fur. So they molt before they're 4 months of age, and at 4 months of age is about the time that they tend to go out in the open ocean and stay out there. So when they are in between that 0 to 4 months age, they do play out in the water sometimes and try and, use their flippers and figure it out, but they're not out there full time. And so before they do spend all their time out there, they get a brand new coat, and then they lose that patch. So it's pretty interesting because it actually usually happens at about 6 months or 6 weeks. So we'll mark them, and in that time span, you can almost see them starting to lose their hair and get a new coat. So sometimes it can be tough to tell if we've marked that animal or they're already shedding that coat of fur.
Right here.
Again, with the first seals, have you ever seen alopecia in first seals?
I have seen a few cases of it out there. We have seen some animals who are bald. We've seen a few albino animals too, actually, which is pretty interesting. But we have seen animals that have splotchy patterns or no fur on a few spots on them. So. Yeah, we actually had one of our first seals, Lou, who's now at the Seattle Aquarium, failed to thrive as a pup because that was exactly what affected him. So he was a pretty mangy looking little guy, but he was handsome in his own way. He had a lot of baldness on his body. He was basically missing all of his guard hair, and so he looked like he was peach in color where he should be brown because all you could see was his underfur. But then when he finally molted for the first time, he came in with a brand new coat of fur and was totally fine. So his failure to thrive was because of his lack of thermoregulation as a pup, and he just needed that chance to get that new coat. So it really depends on when it happens for them. If it happens within that first month and then they get a brand new coat, they're probably good to go. But if it happens when they're out in the open ocean, that can definitely affect them.
So I have a question. So you mentioned briefly all the sorts of things you can look at in fecal samples. I wonder if anyone is looking at the first seal microbiome.
I can't speak to that for sure. Oh, sorry. The question was, there's a lot of things we can learn from poop, and so the question was, is anybody looking at the microbiome in fur seals? I personally haven't really looked into that topic, so I can't speak to that, but microbiomes are definitely sort of a hot topic right now in biology, so I would guess somebody's probably started to look at that.
That's a great question, and that's something I would have discussed if I had a little bit more time. Oh, I'm sorry. Do they have an issue with sunburn? And so we suspect yes, because the age class that we see it mostly in are weanlings, and the weanlings spend a lot of time on shore because they're learning. They're physiologically not yet adept to go on long dives, and they're growing, and so they spend more time on shore to rest. So they do increase their exposure to the sun, and so we do see some irritation and some lesions. We don't know if that's directly related to the exposure of the sun, or if it's from the condition itself and the scratching. One unfortunate thing as to why we don't know an etiology is because a lot of these animals are live and mobile and doing okay and don't warrant us to collect them for any reason. So our sample size of what we can collect from these live animals is very small. We can only collect samples from them for us if the animals die, because that's what we have authorization for. However, there are rehabilitation facilities that do bring in gray seals that may be debilitated for other reasons and happen to have alopecia, and they're collecting samples as well. So that's a good question. Thanks. Maybe Charlie. Oh, question was, how long does influenza, how does the outbreak, how long does it usually last? So for ours, it seemed to be like a 2 to 4 week period, truly, where we saw the peak in mortality. It all depends on the grouping patterns of these seals, and right around that time, the late summer, early fall, they're shifting where they're grouping because of the seasonal changes in it's approaching winter, they're following their food and their grouping patterns to approach any sort of breeding seasons. And so that may affect it. You know, if you're going to have a large grouping of animals for a long period of time, the ability for the virus to be transmitted amongst themselves is great, but I'm not really sure for influenza viruses in general. I think it just depends on the environment, the hosts, and such. Like with humans, we're constantly moving and constantly sharing our germs all over the world. So, Charlie, you have any other? Okay, thanks.
Here's Maggie. Do we need to wrap up at some particular time here?
You guys can do like 2 more.
Okay. 2 more. I saw 2 more. Yes, go.
That's a great question. The influenza that we found, the H3N8, is it zoonotic to humans? So thankfully, that particular strain was not. However, in our lab studies with Columbia, they did show that it had the potential to adapt to mammalian cells. So that one didn't, but that's why we continue to study these viruses, because they do constantly mutate to find the most optimal host. The question was, did we see a difference when we looked at the weanlings in adults with alopecia? Did we see a difference in ectoparasites? Yes. So we didn't get a chance to really assess that, to sample that and look at any ectoparasites in these individuals here in Massachusetts. Ectoparasites have been found in pinnipeds, I think, on the West Coast. Sorry, I didn't have the answer for you.
Okay, we're going to wrap up there. Thanks, everyone.